Use, Juul Labs, Enjoy, Smoke, Vaporesso, Elf Bar. These names might seem familiar, and it's because they're all popular e-cigarette companies. It's known that many adolescents favor e-cigarettes. In fact, 1 in 4 e-cigarette users use one e-cigarette every day. The primary chemical in e-cigarettes, nicotine, has adverse effects on the brain, and its exposure can also cause an individual to get addicted to other products like methamphetamine. In fact, scientists just discovered that a single letter change in your genetic code could determine whether nicotine primes your brain for methamphetamine addiction, and if this effect can vary depending on whether you're male or female. But to truly understand this, we have to first look into how addiction actually works in the brain. When someone takes a drug like nicotine, the brain releases a chemical called dopamine. This happens because nicotine binds to the nicotinic acetylcholine receptors in the brain, kind of like a lock and key mechanism. And then this binding and release of dopamine is what makes you feel alert and rewarded, and can eventually lead to someone becoming dependent on the drug. However, a drug called methamphetamine, or more commonly known as meth, is a little different from nicotine. Meth is often described as being more potent, and although meth causes the release of dopamine, it also transforms the dopamine transporter, so that instead of the dopamine being removed from the system, it's instead transported out with a greater frequency. And so this large increase of dopamine leads to extreme alertness, euphoria, and could even lead to potential neurotoxicity with chronic use. And so with that, welcome to the Translational Addiction Podcast, where we explore the innovative research from the Lotfipour lab at UC Irvine, dedicated to advancing translational addiction science and developing more effective treatment strategies. I'm your host, Ishva Patel. I'm a second-year business administration and biology major at UCI, and I'd also like to welcome our co-host.
Hi, my name is Arshiya, and I'm currently a first-year biomedical engineering student at UC Irvine. Today, we'll be discussing the paper Subchronic Nicotine Exposure Influences Methamphetamine, Self-Administration, and Dopamine Overflow in a Sex and Genotype-Dependent Manner in Humanized chRNA-6 3' UTR SMP Adolescent Rats by Diana Carreño, Antonella Facundo, Anjelica Cardenas, and Shahrdad Lotfipour. To break it down, this study looks at two specific things. The first is how the chRNA-6 gene can impact how exposure to nicotine influences an adolescent's meth intake, depending on whether or not they are male or female. The chRNA-6 gene codes for the alpha-6 subunit of the nicotine acetylcholine receptor. The alpha-6 subunit is a small part of this receptor. In this study, researchers substituted a single nucleotide in the 3' untranslated region of the gene to represent two genotypes, the CC genotype and the GG genotype. The CC genotype refers to rats that are homozygous for cytosine at this site, and the GG refers to rats that are homozygous for guanine at this site. We'll get into exactly how this modification was possible later in this episode, but by looking at these varying genotypes, the researchers could look at what role this genetic difference in the alpha-6 subunit plays on the function of the nicotinic acetylcholine receptor, and overall how an adolescent is more or less likely to get addicted to meth after having taken nicotine. We talked about the lock and key mechanism before, and in this case, the different genotypes could change how the alpha-6 subunit is regulated, altering the way the receptor functions. The researchers wanted to look at how different varieties of this lock could interact with nicotine, and if this initial interaction could shape how it later interacts with meth. If we are treating nicotine like a key, it could make the lock more or less sensitive to the next drug that comes along. But it is not only important to look at the effect of each variant, but also consider when the initial exposure to nicotine was and who the individual is biologically, because this could lead to completely different effects. The second objective is to look at the dopamine overflow in the nucleus accumbens shell after taking meth and how this varies amongst the same genotypes we just mentioned, and whether or not adolescent nicotine exposure plays a role as well. As we have touched upon in previous episodes, the nucleus accumbens shell is a key component of the reward circuitry in the brain. Nicotine and meth significantly shape the dopamine levels in this part of the brain. Considering the fact that this region is directly linked to drug-seeking behavior, it is an important region of the brain for researchers to study to allow them to directly draw a line between the dopamine levels present, or what's chemically happening in the brain, to the behavioral decision, which is how often the rats choose to self-administer meth. By looking at these variables, the researchers can gain more insight into how sex and genotype play a role in co-substance use in adolescence, helping them develop strategic strategies to address and help with substance abuse disorders in the future. So with that, let's take a look at how they actually explored these ideas.
The researchers bred the chRNA-6 3' UTR SNP knock-in rats that were used for the study. The SNP refers to the single nucleotide polymorphism, which we also discussed earlier. But amongst those rats, the juveniles were weaned, meaning that they started becoming self-sufficient in terms of food instead of relying on their mother's milk, on postnatal day 21, which is also 21 days after their birth. And then they were separated by sex and handled by the researchers so they don't fear them and are more comfortable with them. They were placed in groups where they had access to food and water and lived on a 12-hour lie-in-dark cycle. Then the drugs that would be injected into the rats were prepared. So firstly, the nicotine was prepared and dissolved in saline. The methamphetamine and carprofen were also dissolved in saline. Equesticin, which is an anesthetic, was prepared in the lab and it contained a mixture of pentobarbital, chloral hydrate, propylene glycol, ethanol, and magnesium sulfate. And lastly, meth, the nicotine, equesticin, and carprofen were all filtered with a sterile filter to remove any bacteria or viruses from the solution. After the drugs were prepared, it was time to prepare the rats. They were anesthetized with the equesticin, and then a chronic catheter was implanted into their right jugular vein. The catheter was used to administer the drugs directly into the animal's bloodstream. And then the rats were given the prepared carprofen to help them recover and prevent infection. And to ensure that the catheter was safe and functional, the researchers also gave the rats a little bit of propofol to ensure that the catheter was inserted properly. Propofol was specifically used for this purpose since it crosses the blood-brain barrier extremely quickly, allowing the rats to feel the effect of the sedative quickly. So if the rat is sedated quickly, it's safe to assume that the catheter was inserted properly. And so this catheter patency was tested not only before the experiment started, but also before and after any administration to ensure that the catheter was working properly. And then on postnatal day 28, the rats were administered their first dose of nicotine. The rats in the nicotine experimental group were given 2 injections of nicotine, which were spaced 1 minute apart daily for 4 days, while the saline control group was given the first 2 saline injections spaced 1 minute apart for 4 days. The dose the researchers used per day, which was 0.6 milligrams per kilogram, is the same amount of nicotine someone intakes if they're using around 1 to 2 cigarettes a day. And then the specific time of 2 injections a day for 4 days is what the researchers used to model adolescents who initiate using nicotine early in life. So once these treatments ended after the 4th day or postnatal day 31, the rats were tested in self-administration chambers with 2 nose poke holes from postnatal day 32 to 36. The nose poke holes have an infrared sensor to see how often the rats poke their nose through the hole. And from the 2 nose poke holes, one is the control and the other is the reinforcement hole. This means that nothing happens when the rats poke their nose through the control hole, but when they poke their nose through the reinforcement hole, they are self-administered methamphetamine.
To measure dopamine levels in the nucleus accumbens, the researchers first performed surgery on the rats. They drilled into the skull at a very specific coordinate that corresponded to the nucleus accumbens shell, which is a brain region involved in reward and addiction. They then implanted a cranial guide cannula about 2 millimeters above the target area. You can think of this cannula as a small tube that would later allow them to insert the equipment needed to collect fluid from that brain region. The cannula was secured in place using dental acrylic. After the surgery, the animals were given 3 days to recover. During this time, the researchers handled them daily and made sure their catheters were flushed to keep everything functioning properly. On postnatal day 28, the rats received a pre-treatment of either saline or nicotine. The actual dopamine measurement experiment began on postnatal day 31. At that point, the researchers inserted a 2 millimeter microdialysis probe through the guide cannula. This probe has a special membrane that allows small molecules like dopamine to pass through. As dopamine and other chemicals moved across the membrane, the probe collected samples from the fluid surrounding the brain cells. Before giving the rats any drugs, the researchers collected samples for about 60 minutes after the brain had stabilized. This allowed them to establish a baseline dopamine level, meaning the normal dopamine level before the drug treatments. Then the drug test began. First, the rats were given a saline injection. After 100 minutes, they were given nicotine. Then, 220 minutes after that, they were given methamphetamine. Once all of the fluid samples were collected, the researchers injected them into the HPLC system, which stands for High Performance Liquid Chromatography. This system separates the different chemicals in the sample. After the chemicals were separated, an electrochemical detector was used to measure dopamine. This works because dopamine can react with electricity, so the detector picks up an electric signal that can be converted into a dopamine measurement. Using this data, the researchers could compare dopamine levels at baseline to dopamine levels after each drug treatment. This allowed them to see how nicotine pretreatment changed the brain's dopamine response to nicotine and methamphetamine. And what they found was really interesting, so I'll pass it off to Ishva to walk us through the results of the study.
So, what does all of this actually mean? The biggest takeaway was that adolescent nicotine exposure changed how the rats later responded to meth. But those effects depended heavily on both sex and genotype. In other words, nicotine did not affect every brain the same way. So, starting with the female rats, the alpha 6 CC females that were pretreated with nicotine showed strong and consistent meth self administration across all 5 testing days. These rats repeatedly preferred the reinforced nose poke hole, meaning they actively learned to seek out the meth reward. Interestingly, saline treated alpha 6 GG females also showed strong meth seeking behavior, even without prior nicotine exposure. And these females developed this preference later in the experiment, mainly around days 4 and 5. So this suggests that the CHRNA6 genotype influenced not only whether the rats showed drug-seeking behavior, but also how quickly that behavior developed after nicotine exposure. The male rats showed a different pattern. In males, nicotine pretreatment itself appeared to have the strongest effect regardless of genotype. So both of the alpha 6 CC and the alpha 6 GG nicotine-treated males responded significantly more for meth compared to saline-treated males, especially during days 2 through 4. So while female outcomes were strongly shaped by the interactions between sex and genotype, male outcomes seem to be more influenced more broadly by the nicotine exposure itself.
To connect these findings to what was chemically happening in the nucleus accumbens, the researchers found that under acute conditions, alpha 6 CC female rats showed significantly greater dopamine overflow after both nicotine and meth exposure compared to alpha 6 GG females. This suggests that the reward circuitry in alpha 6 CC females responded more strongly to these drugs. However, after repeated nicotine exposure over time, these genotype differences became less pronounced. This suggests that chronic nicotine exposure may alter the extent to which genotype influences dopamine signaling. In male rats, the pattern was somewhat reversed. Acute meth exposure produced greater dopamine overflow in alpha 6 GG males compared to alpha 6 CC males. Unlike in females, nicotine exposure itself did not produce major genotype-specific differences in dopamine releases in males. The researchers also measured dopamine metabolites called DOPAC and HVA. These chemicals are produced when dopamine is broken down, so they provide insight into overall dopamine activity and metabolism in the brain. Female alpha 6 CC rats showed significantly higher DOPAC and HVA responses after nicotine and meth exposure compared to alpha 6 GG females, suggesting that dopamine processing differed substantially between the two genotypes in females. In males, these metabolite differences were much smaller and appeared mainly after repeated meth exposure.
So, taken together, these findings show that a single nucleotide variation in the CHRNA6 gene can significantly influence how adolescent nicotine exposure affects both the later meth-seeking behavior and dopamine signaling in the brain. And more broadly, the study highlights how addiction vulnerability is shaped by multiple interesting factors, including genetics, sex, developmental timing, and prior drug exposure. So even small genetic differences can meaningfully change how the brain responds to substances like nicotine and meth during adolescence.
To recap, this study showed that adolescent nicotine exposure can change later responses to meth, but those effects depend heavily on both sex and CHRNA6 genotype. Female alpha 6 CC rats showed stronger meth self administration and greater dopamine overflow after nicotine and meth exposure, while male rats showed a different pattern where nicotine exposure itself had a large overall effect regardless of genotype. The dopamine metabolite findings including DOPAC and HVA, also suggests that genotype may influence how dopamine is processed and regulated in the brain. Since dopamine signaling is closely tied to reward and reinforcement, these differences may help explain why certain groups were more vulnerable to later meth-seeking behaviors. The researchers suggest that alpha 6 containing nicotinic acetylcholine receptors and the dopamine transporter may play important roles in these effects, although the exact mechanism still needs further investigation. To build off of this research, future studies could directly measure dopamine release during active self-administration behaviors, examine how these genetic differences affect natural rewards like food, and further explore how repeated nicotine exposure alters dopamine signaling over time. And that's all for today.
If you're interested in learning more about translational addiction science, follow us on Instagram at Translational Addiction Podcast for updates, and check out the infographics and papers on the Lotfipour Lab website linked in the show notes. If you or someone you know is struggling with addiction, you are not alone. Please contact your healthcare provider or refer to the resources linked in the show notes. The Translational Addiction Podcast is a production of the Lotfipour Lab at UC Irvine, exploring how to understand addiction and develop effective treatments. See you next time, and thank you for listening.